bioinformatics from the University of Kansas, US, with precious role including senior associate project at Cognizant, data scientist at Flipkart, director and head at Novartis Institute of Biomedical and Research. Dr. Mukherjee has experience across various sections. His talk titled Supercharging Traditional Bioinformatics Problem with Deep Learning and AI is anticipated to draw upon his expertise in data science and bioinformatics, offering insights into innovative approaches to address traditional challenges in this field. So I welcome you, sir, for your talk.
Thank you very much. Let me share my screen. Let me know once you can see it. Okay. Is it good? Can you see my screen? And can you hear me?
Yes, sir, we can hear you.
Perfect. Thank you. First of all, thanks guys for inviting me for this. Super excited to be presenting here. Yeah, so my topic is supercharging traditional bioinformatics problems with deep learning and AI. Right. So I was introduced, I started off obviously as a bioinformatician who later moved on to becoming a data scientist or an AI researcher or something, right? So first of all, this is meant as a pun, but all views here are my own, does not represent any organization I work with, because science is cut across organizations, right? What we're going to talk about, we're going to have a little bit of introduction, and we're going to deep dive into 3 different use cases that I am working on, have worked on, and because I come from the corporate world, also maybe I'll leave you with something that beyond the hype of data science and AI, what it takes in the real world, right, to actually do AI. I'll start with a very famous diagram. It's called the Drew Conway's data science Venn diagram, where it basically says that to become a data scientist or data science is the intersection of 3 different areas: maths and stats, hacking skills, and substantiative experience or expertise. This is very true, and the more I've been in this field for quite some time now, and I can say that I agree with this 100%. Why math? Because it is a language of science, right? It's the language with which nature speaks to us. Why hacking skills? It's because it enables problem solving efficiently. We don't have a lot of time. And why substantive expertise is because without this domain expertise or this knowledge, we end up solving our own problems rather than God's problems, right? And a true model, a data science model, an ML model, etc., should be data agnostic. It should be, and a data science scientist should often be domain agnostic, right? You should be able to pick up that substantive experience. Also, I'm very scared of these people. People who have hacking skills and the domain expertise but have no knowledge of math and stats. I generally stay away from them as far as possible because they are a danger zone, right? Okay, so what is bioinformatics? Now, this is how I have evolved. This is my own journey and how I have evolved. So if somebody asked me during my undergrads, right, that what is bioinformatics, uh, probably you would have found me biting my tongue because I was not really sure what bioinformatics is. I would tell you, it's something to do with biology that you do with computers, literally. That's all my understanding as an undergrad of bioinformatics was. During my PhD, that's when I would have defined it as using math, stats, physics, chemistry to a certain extent to understand biological systems using the medium of computing, right, through programming languages or software of some kind. Today, though, I sometimes define it as using principles of data science to comprehend, analyze, and predict biological phenomena at the system level. Right, so it's more and more the world is evolving to a system understanding of biology, and bioinformatics or data science is one of those mechanisms which can help us reach that comprehension of the system level faster. Right, that's what I define. Now, I'll give you another case of knowledge evolution for myself. Right now, during my undergrad, again, I knew that why I say actually that models should be domain agnostic, data agnostic, time agnostic also as well, I guess. So as an undergrad, all I knew that to align two similar protein sequences, we needed to use Needleman-Wunsch dynamic programming. Now, that's all. That's all I knew at that point, literally. After the first month of my PhD, when I went to US, so I was asked to write a code during my course using any language of your choice for using Needleman-Wunsch algorithm. So they asked us to write that actual code and align to protein sequences, not downloaded code, but you have to write your own. Then, sometime during my PhD, I published my first paper on dynamic programming to align to protein complex structures. During postdoc, I went back to dynamic programming again to align to sugar structures. Right? It's called GSLI. Then I went for my first job. When I at my first job, I was using dynamic programming again to align to peptide scaffolds to identify drug-like scaffolds in the PDB library. From drug discovery in my early life, I moved into Flipkart doing supply chain and forecasting and things like that. But the funny part was, I again used Needleman-Wunsch dynamic programming, but this time to do product lifecycle forecasting. For example, how a mobile phone is going to launch, how it is going to be during a maturity period, and is it going to die, right? And I use Needleman-Wunsch dynamic programming for that. At Novartis, I used to work a lot on medical informatics as well, right? And I used to work on electronic health records of patients. And I used, at that time, neural networks is coming in fast and strong. And I used Siamese, a particular type of neural network called the Siamese twin network to do dynamic time warping, which is actually very, very similar to Needleman-Wunsch, to align the electronic health records of multiple patients together, right? So that's how my knowledge has evolved, and that is the basis of many things I'm going to talk about today, right? Now, AI and data science has been coming up very thick and fast, and everybody's been talking about it, but and everybody wants to do it. But I keep saying that, ask not what your data can do for you, ask what you can do with your data. And certainly the future does belong to those who have data for it today. Because data is a new gold, right? Cool. I will then start deep diving into very similar, not Needleman-Wunsch, but transformers, and how to capture long tail of biomedical knowledge. Right? What is this? As we're about to publish this, so we call it drug AI. Drug AI basically takes multiple documents, 35 million publications, uh, grants. It takes patents into account, clinical trials, 96,000 clinical trials, all kind of biomedical literature, basically, right? And integrates this vast corpus. It's a really large corpus into a knowledge graph. We are basically taking all these articles, breaking it up into three entities, either a disease, a target, a drug target, and a compound, a chemical compound which acts as a drug. Right? So we broke it up into these three parts. And we built what is called a knowledge graph out of it. Right? How did we do it? We took LLMs, large language models. Uh, of course, they are again ChatGPT and Google Bard, and all of this are coming in extremely, extremely fast. So LLMs, of course, are an extremely powerful tool, and we use that to mine the biomedical literature and convert it into a knowledge graph. Right? We did not use ChatGPT or something. I'll come to what we did. What we did is something called named entity recognition of our data sources. We broke it up into biomedical entities. In this case, we just took three entities. We came up with around 5,100 diseases, around 100,000 genes, including non-human species like bacteria, and 25,000 drug compounds from Campbell, right? And we extracted the relationship between these entities, and we connected them together using into a knowledge graph. So today in that drug AI, we have around 2.5 million disease and gene relationships, right? Another 680,000 gene to gene relationships, tied somewhat, right? Gene drug relationships. So we basically took biomedical literature and broke it up into a graph structured data instead of a bunch of documents, right? Why did we do it? We'll come to that, but before that, just a little bit of deep dive on how we did it. So we manually labeled output annotations of all biomedical entities, right, for some. And we took mesh ontologies and other gene ontologies, etc. We used a particular model called Kazoo or Burn2, right? It's a large language model. And then we identified 8 entity types, including gene, drug, and diseases, right? And then semantically and contextually, so for semantic and contextual understanding, we use BERT-LES for multi-level prediction, right? Uh, and that's what we had. Once we broke it up, then the next thing we did is look at the question of why are we doing this? Now, if you look at biomedical literature and you're searching for biomedical literature, the problem that often happens is something called an information overload, right? There's so much information out there that it's super difficult to ask or answer a question. Suppose somebody has a very simple question. What are the known drug targets for treating asthma? Or what is the relationship between losartan and hypertension, right? Very simple questions. Now, there are two ways of doing it. Till late, it was only one. It was only a search engine that you can get, and you can either get links to research articles on that, and then you have to go and read it, or you need to, you can get review articles sometimes, and you need to go and read the review articles and move back and look at other research articles that it cites, etc., etc. The latest way of doing it, of answering these questions, is what I call LLMs, right? The large language model, the chat GPTs and MedPalms of the world, the GPT-4s of the world. There are two problems with large language models. One is it hallucinates a lot. So it talks rubbish most of the time, right? And one way of keeping it to context is something called a RAG, or a retrieval augmented generation, right? Now, retrieval augmented generation, first of all, has two advantages. One is it is up to date answers, it's not depending on the pre training date. The second thing, it typically keeps things in context. But again, what happens is it suffers from this information overload problem, which we are trying to solve, which is the topic of the study, right? For LLM plus RAG, how do you solve the information overload problem, right? So this is the drug AI knowledge graph, and this is what we are trying to do. So when you have a biomedical corpus like the PubMed, and suppose you've not broken it up into a knowledge graph. Today, what a GPT-4 or an LLM does is creates a vector database which contains embeddings of all your documents, or a latent representation, however you call it, and it stores it in a vector database. Once a user asks a question, like as of today, which are the most promising drug target for diabetes, it embeds the question. Uh, then, basically, according to cosine distance or some similarity matrix, searches through the vector database to find out documents which are most similar in embedding space to the question that is being asked, right? And that's your retrieval. That's your option one, that is your embedding similarity. The second one that we are saying is a knowledge graph approach, where we have broken it up into different entities. What's different? So in B, you will see that, so if question embedding is pink, it has drawing this circle around it, or of a particular radius. And any article that falls within that particular threshold are retrieved, and the generation happens basis that. Using a knowledge graph, what happens is, instead of embedding the question, we first identify the entities that the question talks about and the relationship uh, that are being talked about. So those are the two pink circles on the C diagram, right? And then what it does, it identifies the shortest path linking various different identity entities, right? And everything that's connected to it, all the articles that are connected and mention these particular entities, the yellow ones, for example, and the relationships that they're talking about are extracted. So these are two different ways of doing it. The third one is just combining the two together. Right? Why do this? First of all, when you're doing search, you know, recency, so citation, because in the page rank type of algorithm, recency is important, but citations and how many people have clicked on it is also very important, which then has a slight problem. The recent articles have lesser citations, but more likely to contain newer discoveries, right? So we use the Pareto ranking, which balances the two out between recency and number of citations, etc., so that retrieval scores are better, literally. Now, this is only 5 therapeutic areas. We've done it for the whole thing. So these dots over here are various documents, literally, the various documents or various articles on either of five areas of this thing, and we mapped it onto the latent space. There's a new map diagram of the embeddings of each of these articles. So on the one on the left is according to therapeutic area, and one on the right is according to your heredity type. So your disease, your drug, and your gene kind of embeddings. While on the left you have these various different embedding types. Now to test our method, what we did is we use a particular set that is already available where answers are known, right? And the right answers are known and the retrieval answers are known. So if you look at it, right? So we did it with two ways. We did the retrieval in two ways. So either we used embedding similarity, which is a typical RAG that happens in a ChatGPT interface or anything of that sort. And the second one was augmented that, or not augmented that, we used a knowledge graph to do the retrieval and then used any of the large language models to do the generation. And the third one was you combine the two of it, right? Now, if you look at the recall at the rate and precision at the rate k, right, you will find that the knowledge graph approach beats the embedding similarity approach by a large margin, right? However, another important thing that came out, which was surprising, is there was a lot of non-overlap between the regions it was extracting, right? So because the hybrid model actually outperformed both of them individually. Okay, why did that happen? Uh, so let's look at the embedding space again, right? So the question embedding is the one in the blue bold X over there. And the ones with the arrows, the areas where we were talking about, what are the known drug targets for treating asthma? This was a particular question. The answers were all over the space because when you were embedding it, it was not necessarily close, but it was all over the place, right? So if you look at the entity embedding or the question embedding, so the ones again, like the arrows are the right answers. That's where the right answers actually are in the latent space. Now, when you used retrieval embedding, it's simple retrieval, embedding similarity based retrieval. Given the question, it did identify the right questions, but the blue area, right, the shaded blue area is where your typical RAG or retrieval of a large language model is restricted towards. When we used the knowledge graph, we found that while there was an overlap between the two of them, it encompassed a much larger area, right? So it was able to tap into the long tail of biomedical information. Hence, the answers were more complete, literally, because there's an information overload. There's a lot of information out there. Finding out what is good is a problem. So, and typically, when you're using something like very simple retrieval like similarity, then it is not able to get into the long tail of this biomedical information. When you use the knowledge graph, you could again suddenly extract a far more complete answer, right? So that's the first one that I want to talk about. This is a simple informatics, biomedical informatics kind of problem that we have a lot of publications. Everybody's publishing like crazy, right? Nobody can keep up. How can we make sense of it? Cool. Okay, another few quotes. I love to do this in general. We use data whenever possible. It's always possible, right? So that's what I keep telling many of when I am at work, literally, because I work in traditional organizations, which are moving from a very, very non-data-based approach to a data-based approach. So these are some of the quotes I often use. Next one that I want to deep dive on. Again, very similar in terms of architecture, in terms of technology, but a very different question that I'm trying to answer. That how do you use large language models in drug target identification? This time, not from literature, but from uh, actual omics data, right? So, and this is all single cell based. Why should we study single cells? First of all, because the body is made up of different kinds of cells, skin cells, immune cells, brain cells, etc. And various diseases are actually restricted to particular cell types. For example, autism has something to do with neuronal cells only, or muscular dystrophy is something to do with skeletal muscle cells only. So when we study any disease, right, we generally end up we don't often, but we should study particular cell types, right? That's what is key. And they're the key between the genotype to phenotype, right? And essential for functional dissection. So we're going to talk about single cell studies only. Now, if you look at biological regulation, first of all, it is extremely complex, right? And it is extremely complex because there are many, many different layers of regulation, and the space of regulation is extremely large. Uh, as a context, the entire internet today is roughly 10 to the power 13 tokens or words, right? Uh, the number of expression levels within cells in the human body is expected to be or estimated to be somewhere close to 10 to the power 60, right? Number of regulatory mechanisms that are possible. So it's extremely complex. So what we did, or we are trying to do, it's not done yet. So we basically said, okay, let me break it up into 6 different layers. So if you think of a graph, it's like a layered graph where every layer is a particular state or particular scale in the biological scale. So first is the genome scale, which is your DNA. This also contains your epigenetic information like methylation and histone modification, etc. The second one is your RNA scale or the transcriptome scale where the expression is provided, right? And epigenetics again can be a further sublayer in this one. Then you have the proteome scale, which basically represents protein-protein interactions, protein RNA interactions, protein DNA interactions, etc. Then these proteins which are interacting typically then form a pathway, which is your pathway scale. So co-memberships are in a particular pathway represented by links as well. The multiple different pathways then come together to form a biological process, let's say metabolism or whatever. And finally, all these processes, multiple processes come together to produce a particular phenotype. Right? So there are 6 scales that at least we are tackling it from. And diseases, and if you look at these different layers, right, and if you brought it out into a graph, this is a rare map of rare gastroenterological diseases, right? You can find links to it at various different places. So, coessential genes, for example, is one where Schaumann-Diamond syndrome, the cause of Schaumann-Diamond syndrome is embedded. So ribosome biogenesis, there's a problem with ribosome biogenesis in that particular one, right? Esophagus co-expression. So what is happening in the esophagus, in which the genes are being expressed and which are not, is often where you find the underlying cause to esophageal, gastric, and colorectal cancers, right? Similarly, the protein-protein level, pancreatic cancer, today we've been able to trace down to the protein-protein level, right? Pathway memberships again. So there are different. This is just a sampling of it, just for representation. In kind of this way, what we're trying to do, we're trying to understand the cause of underlying diseases, right? And we're trying to do. What we're trying to do is basically understand where a therapeutic intervention should be designed towards, right? Which layer? Now, silicon, so now we have 6 different scales, right? So there is a lot of work that's been going on for a long time in these individual omic spaces, from the genomic space, from the transcriptomic space, etc. What we are trying to do by breaking it up into the 6 different scales is to kind of take a multi-omics kind of approach. What is multi-omics? Why should we do multi-omics? Because first of all, if you have to understand causality, it is sometimes not sufficient to understand only one layer or one scale. You should try to link it to all the other scales that are above it or below it, right? And what we're trying to do in the target ID space is understand or discover novel cell types. We're trying to understand tissue and tumor heterogeneity, generate more complete atlases of the cell, do biomarker discovery, and then understand complex diseases and pathways and networks of regulation, right? And what are we doing? So we are taking these individual omics profiles and we are taking prior knowledge like published atlases, etc. And we take a multi-stage or a multi-modal analytical approach, and we are taking 3 different routes. So we're taking machine learning based methods like unsupervised learning or deep learning, which I'm going to talk about somewhat. We are using also diffusion propagation methods and causality and causal inferencing kind of models, right? And we're trying to get this connection between these different scales, L1, L2, L3, and how is regulation happening in a particular disease, right? So again, we come back to large language models at this point. Large language models, as what it's famous for right now, is understanding text, right? But text is just one modality. And as far as I am concerned, text is nothing but a sequence of words. And our DNA or an RNA or a network is nothing also but a sequence of genes and tokens. Right? The other thing that LLMs have brought about is this concept of transfer learning. So, standard approach to machine learning or deep learning has always been that we have labeled task-specific training data, right? We use supervised training of the model and we get a trained model and then we inference it or use predictions for something else. So, what transfer learning or this expressive power of these large models does is it says that look, if you strain on a very large data set, extremely large, of course, it's extremely expensive to do it, and and of course, you can't find annotated or supervised data for it, so you kind of self-supervise it by doing kind of fill in the blanks or predicting the next word out or next token out or next expression out, etc. And then if you train on it, so this is, for example, a particular model that came out last year, so in the middle of last year, 2023, we call it a single cell GPT. There's another one called Gene Former, for example, right? What they do is we take these atlases of large atlases across cell lines, different cell lines. You pre-train a model with input embeddings and master attention transformers. And what you do so you get this base or foundation model. Then you take new data that is being generated in a pharma company, for example, and which are very specific to particular diseases or particular processes, right? And then you fine tune this foundation model. And now, model is what do you mean by fine tune? The model is nothing but a set of weights at the end of the day. These neural networks or transformers, nothing but a set of weights. That's all there is to it. So when you pre-train, you start from a random set of weights, and through your pre-training, the weights are learned. And when you're doing a fine tune, instead of starting from a random set of weights, you start from the weights of a foundation model, right? And we fine tune it, and then post that we can do a lot of things. Clustering, for example, can be done to do cell type annotation. Batch corrections can be done to understand gene network inference, right? Or multi-omic integration can be done. And finally, we want to do perturbation predictions. What is perturbation prediction? That you have let's come to it, right? So we're trying to do four things with it. One is to do target ID with gene networks. So we're trying to get this or derive this gene regulatory networks, right? Uh, and then say that, okay, just do typical network analysis and say, okay, these are the ones where network exhaustivity is the highest and benefits most from high throughput. If we pick this out, there's a good chance that a disease can be cured, a therapeutic intervention can happen at that particular node in the network. The second is what we're trying to do is do pseudotime ordering. This is at a cell embedding level. This is not at the gene embedding level, right? So a cell goes from healthy to disease type 1 or 2, and it forms a particular or it follows a particular pathway. So by doing expression analysis at different stages of the cell, you can kind of map how does a healthy cell move towards disease type 1 or disease type 2, right? And we're trying to understand that with high resolution cell omics data. Third one is called similarity modeling. This is again gene embeddings. For example, there's an embedding of the gastrointestinal space that we're talking about, right? Uh, Everything that's in orange has at least one clinical trial. So it's a validated target, right? It's a validated protein target mostly. And there are clinical trials either completed or ongoing in these particular ones. But very close to these things in terms of gene embeddings are other drugs or other targets against which no drug has been designed, right? So can those also be used as possible drug targets? That's our endeavor in this target ID with similarity modeling. The fourth one is again from a cell embedding perspective. And these are to do with in silico perturbations, right? We are trying to do virtual knockout experiments. So So in cardiomyopathy, and this is a particular one from cardiomyopathy. So you can, once you have the embeddings of the cells, you can classify them into either non-failing, which is your healthy ones, or hypertrophic cardiomyopathy or dilated cardiomyopathy, right? And what you're doing is all those gray or the circles are particular genes in that particular cell, right? If you knock out one particular one, does it now start classify or move towards being hypertrophic or dilated? And we're trying to see, okay, which of these genes, as far as the model is concerned, are the most important ones according to which it classifies a particular cell type to be the non-failing or a disease one like hypertrophic and dilated. And the whole hypothesis is because the model understands these cells from an embedding perspective and a gene perspective, it identifies the genes by doing these knockout experiments. You can identify the genes which are actually responsible for a particular disease. This is the four types of things we are trying to do. So again, I want to say that there's a lot of work that's going on here, and all if you look at it, so gene former is one particular model that is super popular at the moment. It's a BERT-like autoencoder, and it's an encoder only model. It's a large language model which is used or trained to do these kind of gene and cell embedding. SCGPT is another one, and this is a decoder. It's very GPT-like. It's a GPT-like decoder. Then you have other ones like depth maps, which is a graph neural network, and it's an autoencoder framework, so it's got both the encoder and the decoder. And then there's DSpin, which is much more closer to what has been done earlier, which is more probabilistic, which is done with Monte Carlo sampling. Right? And we're trying to see how we can combine the best elements of all of these kind of models, right, to drug target identifications. Okay, now the only thing, so I started off like as a bioinformatician doing protein modeling, right, protein structure prediction and stuff like that, and I do have various different things. And so I often say that the only thing that interferes with my data is my education. So I keep going back to okay, proteins and stuff like that. When really the same type of model, same type of technology can be used across of answering various different types. So in that mode, I'm going to talk about a very similar model which is being used for an extremely different purpose. And this is in clinical trials actually, and this is for detecting deep brain atrophy when neurodegeneration happens in Alzheimer's and Parkinson's and multiple sclerosis with 3D vision transformers. Very similar architecturally to a gene former or a BERT or any of GPT, so on and so forth, but a completely different application, right? So segmentation is a particular process where we try to understand in an image which area belongs to which particular subtype, right? So in this particular case, we are especially interested in something called the deep brain. An atrophy in the deep brain, and there are multiple different parts of the deep brain, the caudate pallidus, etc., the amygdala, the pallidum, the putamen, etc., where atrophy happens, and this atrophy is often or correlated or causative of various neurodegenerative disorders. For example, in MS, no, it's known that subcortical structures are affected, and there's an association of DGM atrophy with memory impairment. Similarly, in Huntington's, the severe atrophy has been absorbed. Parkinson's to iron accumulation in the DGM leads to protein deposit formation and bradykinesia, which is a trembling that you see in Parkinson's, right? Alzheimer's, multiple different parts of the deep brain are affected. Cerebral palsy, the basal ganglia is affected. The current challenge in MRIs, and these are 3D MRIs, right? MRIs are 3D images. Quantifying this DGM is extremely laborious. It takes a long time. It requires extremely specialized skills, and is super difficult to do. And there are atlas-based methods which do handle, which are used to do it. It's laborious, but it does not handle deviations in brain anatomy caused by injury or very well, right? The traditional machine learning, deep learning have shown promise, but it does not generalize. Yeah, you have fun. So you always lack annotated data sets. So traditional machine learning does not work because there are lack of large annotated data sets, right? Now, if you could do automated DGM segmentation, so deep gray matter segmentation, right? What you could do is do volumetry. So see if the volume of the subcortical gray matter is decreasing or not. You can do tractography, by which MRI can be used to track, how degeneration is happening. Uh, get more information about the texture. For example, when protein iron accumulation is happening in Parkinson's, the texture of the DGM changes. Can you track that? Does the shape change of the various different parts of the DGM, etc., etc. So if you could do it, and it's extremely difficult to do, then it has extremely valuable actual real life applications, right? Now, again, we start what we are doing. This is the overall approach. So we are taking a multimodal MRI dataset. Now, MRI is multimodal, so there are various different modes of an MRI. There's T1, T2, flare, gadolinium T1, diffusion tensors, etc. And each of them tell very different things or capture very different information. So it's often difficult to combine them together because it's just talking in a different language. Right? So we're doing a pre-processing of it first of all, and then passing it through something called a vision transformer. But it's not a typical vision transformer. It's a vision transformer combined with a more older architecture called convolution neural networks, right? And then we're getting again this foundation model and some semantic priors that are being extracted from it, right? And once we have this foundation model, and then for a Parkinson's or an Alzheimer's or a multiple sclerosis, we can do, because the base weights are already set. Now, with even lower number of annotated data sets, we can do deep supervised fine tuning and do segmentation and do volumetry or textural analysis in Parkinson's or Alzheimer's or something like that. Now, hybrid transformers. Why we're using hybrid transformers? We have experimented. It's also been published that they generally outperform transformer only methods for some reason. Especially when there's imbalanced class segmentations. If we look at MRIs, the number of healthy MRIs is far larger than the one with DGM atrophy. So typical class imbalance problem, right? And hybrid methods are generally much better. Uh, Another thing that we are trying to do, but we're not going to try, which is augmenting with text-based input. So instead of using actual labels, we can get a report, right, of a doctor saying that, look, deep brain atrophy is happening over here, and this is what explains to it. How can we then tie the text of that and with the image of the MRI? Right? These are certain things we're trying. And now, even in these embedded or these hybrid transformer models of sorts, right, there are numerous different ways of combining them together and achieves different kinds of results, right? Now, remember what again the theme is that it's a transformer. The transformers, I'm not working with the core architecture of the transformer, whether the transformer is the best architecture possible or not, and doesn't generalize or does it memorize, not getting into that. But for a particular application, like either gene target identification, drug target identification, or for something like this, where we're trying to segment images and understand deep brain atrophy, you can use these technologies that exist, like a transformer, combine them together with older architectures or use it in very judicious ways and get results that maybe work, and you can keep experimenting on it, right? So there's many different types of architectures are possible. We are experimenting on those with these particular transformers. Now, what we see again is pure transformer architectures are quite competitive actually. But the segmentation results are average, especially when it comes to imbalanced data sets. Right? Uh, why I guess these things are so good in general, because there are mechanisms within the transformer to preserve this high resolution informer compared to CNNs, improves performance quite a bit, right? Hybrid architectures, on the other hand, tend to have very fluctuating results, but they invariably somehow outperform the pure CNNs or the pure transformers, right? Within that also, when I'm using a VIT or a vision transformer as a decoder, right, in a GPT style decoder, the performance is relatively poor, even actually worse than pure methods. But when I'm using it as an encoder, then, then the performance is actually much better. However, the best thing that happens, which is, and we still haven't figured out why, is when I'm using a CNN-based encoder and a CNN-based decoder with a transformer in the middle, that's when the performances are the best in this particular thing. And we've also tried it on tumor segmentation, not just in deep brain atrophy. And the results are super similar, right? Cool. Uh, Now, I will come to the real world, right? What it takes to be a data scientist in the corporate world. Now, unfortunately, in the corporate world, in the real world, good data is actually a respectable optical illusion. It ruins many a data scientist. So while the AI and data science seems like a fairy tale, which is on the left with Snow White and Seven Dwarfs, the reality is more towards the right, right? Kids running around and it's crazy at times, right? In the corporate world. There are three or four different things that I want you guys to take home as what matters in the real world. One is that data is equally, if not more, as important as an algorithm, right? The completeness of the data, the timeliness of the data, how valid it is the data anymore, right? Is it integrated? Is connected together, the different data sets? How unique is it, or is there a lot of redundancy in the data set? Redundancy is good, but redundancy, too much redundancy, again, not very good. And how consistent is the data, right? So these are extremely important problems that a data scientist, an AI researcher in the corporate world has to face every day, right? Second thing is, guys, the mathematical underpinnings of machine learning and deep learning are sacrosanct and cannot be ignored, right? I interview a lot of people. And I end up rejecting more than I actually select. And the problem is, you know, their mathematical understanding often tends to be poor. And look, maths is the so at the end of the day, any machine learning problem or any data is nothing but points in geometric space. You call it embeddings, you call it encodings, you call it whatever, but these are just features in a geometric space. These different features come together and form a landscape, right? And all that a machine learning algorithm is trying to do is find the optimal path in that complex landscape to reach a particular point. And to traverse that landscape, the vehicle that machine learning uses is linear algebra, right? Nothing fancy, but linear algebra is sufficient to do optimization, reach that particular space. Now, if you understand these particular thing and keep understanding this, that's when I feel that you can be successful as a data scientist in AI in the real world, right? Simply knowing how to run a model or train a model is not sufficient if do not understand the mathematical underpinnings of machine learning. Fourth, ML models are useless in driving business outcomes unless operationalized. For example, I see this on the camera, I see the whiteboard with a sudo Docker and I can't see the rest. I was so happy seeing that because the fact that Dockers are being used to do it, a simple model is useless because it's not going to be running at scale. So Docker containers are important. CD pipelines, code deployment pipelines are important. Experiment logging and monitoring are extremely important uh, in the real world in the corporate life. So this entire cluster. So if you do not operationalize your models, typically they never see the light of the day and no actual impact, whether it be business impact or big scientific impact, actually happen. Right? Finally, despite everything that you see and hear on LinkedIn and all these different magazines most business leaders do not understand any machine learning, right? They say what they say for lip service, but you as a data scientist and AI researcher, or me as a data scientist and AI researcher, I have found that it is extremely important to be an effective storyteller. Because unless I am a really good storyteller, it is impossible to make the leaders, the budget holders, etc., of how the things are working and how it will help solve a particular business problem. So you have to follow a very, very concrete way of getting models in pipelines to actually make business impact, right? And on the way, along the way, as you story tell, your audience's knowledge has to be enriched, which then in turn increases the likelihood of that person to act. Correct? Cool. With that, I'm going to stop and pause for any questions.
Thank you, sir, for your valuable talk. If anybody want to ask any question, they can ask.
Hello, can you hear me?
Yep, I can.
Yeah, so I have a small question. Like, I heard that there's a topic about decision intelligence, and some work is going on that domain. So, how LLM can help to do that?
Oh, so again, depends on which domain you're working on. Really, see, the reality is LLMs do not think. LLMs have memorized, has seen a lot of information, and it has kind of understood that in a particular situation, these are the most common ways and things happen, and people approach it, or the most common decisions that are taken, and these are the likely outcomes. So it then says that, okay, use this particular way of doing it because that's what has shown outcome. So it is not really thinking and making a decision on what to do, but rather it is summarizing a lot of instances and situations it has seen, and then summarizing that and bringing it in front of you, right? That's a first point in it. So it does help, but from more from a summarization and what has been observed and a search engine perspective. The second thing is keeping it in context. Keeping LLMs in context is supremely important. Otherwise, they move around a lot, and then what it throws out is basically junk most of the time. And there are many ways of constraining an LLM. Now, one is obviously prompt engineering, but prompt engineering suffers from bias. So if you are looking at decisions making, right, and you're trying to prompt LLM into something that you feel is right. Your prompts often tend to be biased towards what you feel the decision is supposed to be. So, how to control for the bias is also very important. Uh, there's a third one that is being explored a lot right now, which is called agents. So, instead of using one LLM, you have multiple LLMs which talk to each other, right? And it's almost like a committee of LLMs, and they are together trying to decide on things. So, there are multiple ways to do it. It's an ongoing field of research. Now, I would like to, there's a guy named Andrej Karpathy. Maybe some of you have heard of him. He's the one who, by the way, coined the term prompt engineering, right? In a recent, I think it's there on YouTube, there's a video. And he was the chief data scientist of Tesla, and he was working on autonomous driving, and he's recently moved to OpenAI. So he makes a statement. There are certain technologies that are extremely convenient to do a POC on, and they're lovely POCs, but super difficult to productize, right, and put into actual business applications. LLMs, and especially agency-based LLMs, are one of those where beautiful POCs can be made. It can be a lot of progress is being seen on toy problem statements, but the business impact is and actually solving certain things and in a productized, scalable way, and the lack of chips is not helping, is often very difficult. Does it answer your question?
Yeah, thank you so much for the explanation.
Cool. Any other questions, please?
Excuse me, sir. So thank you for this very informative session. I am Mina, attending the class offline. So my question is, how do you collaborate with clinicians and neuroscientists and imaging specialists to advance research in 3D vision transformers? That's a
Super interesting question. It's a difficult thing to do. One is to, first of all, the first thing that I always tell my team as well, is to fall in love with the problem, not fall in love with your solution. Just because you feel that I can run a vision transformer, which is like a hammer, I'm going to use every problem statement as a nail. If you approach neuroscientists as that, then that's it. They'll stop listening to you. So I try to first talk to the neuroscientists and understand their problem, right? What is exactly their problem? And then the the crux is try to solve the problem, not use a vision transformer for it just because you know how to run one. That's the first thing, right? When you find that you are open, then the scientists also become open, get open. The second part to that question, the second part is, there's a lot to learn from these neuroscientists, right? Yes, at the end of the day, they are the ones who are going to actually develop a drug. And my job as a data scientist and AI specialist is to make their job easier. I want to give them a particular tool, certain ammunition with which they can do their job, right? On the other hand, they can provide a lot of information to me, a lot of their knowledge to me by how they particularly take decisions when it comes to looking at an MRI, uh, which I can then try and incorporate into my models, right? Typical models, when you build a very large generalist model, it answers generalist questions very well, but specific questions are a problem. And incorporating this human in the loop is important. So, active learning is one thing that I often use. So, it's a uh, constant, cycle between models and a human in the loop, verifying the models, understanding the models, etc., etc. So active learning is something I try to follow. And but most importantly, just having an open mind and understanding that look, they are there to do their job, and I'm here to help do their job, right? Or help them do their job. That I think is the crux, being falling in love with the problem statement rather than your solution as a data scientist.
So, sir, I'm from computer science background, pure computer science background. I don't have any more knowledge, like deep knowledge in biology or chemistry or anything like that. So, can you please give me like path towards this bioinformatics? Like,
Look,
from where should I start?
I think we're already in a good place, right? You're at Kharagpur, so that's already a good place to start, right? So, secondly, look, I started off, and I know one of your professors out there who's there, Pralay, who was my lab mate, actually. None of us actually had a very big biology background, right? We went on the job, we went to do our bioinformatics, our degrees, our PhDs, our postdocs, and then slowly got into it. Even now, I don't really understand neuroscience. I never studied neuroscience, right? But when I started getting interested in it, learning it is important, and reading papers is important, and getting to know the basics is important. Just like maths is important when you're trying to be a machine learning practitioner. Similarly, understanding sodium channel blockers and how it works, and how does potassium sodium balances in the cell help neuroscience, etc. Those are important things to know and important things to learn. And once you do that, you'll find that you are able to make sense of what you are doing. And once you do that, I mean, look, read, I guess. Read and practice, and that's the only way of learning, right? I don't come from a traditional biology background, actually, but in fact, I did very poorly during my class 12s in my biology exam.
Okay, sir. Thank you.
No problem. Any other questions, guys?
There is no more questions. Thank you, sir, for your talk.
Perfect. Thank you, guys. I will log off now.
Sir, you can log on.
Perfect. Thank you. And thanks again for inviting me.